Good morning, everyone. This is Pooja, and welcome to my channel. Struggling to download FASTA sequences from NCBI? Don't worry, today I will show you the easiest step-by-step method in just 2 minutes. So, step first, go to the official NCBI website. The National Center for Biotechnology Information. This is the website where database used worldwide for biological data. In the search bar, select nucleotide if you want DNA sequences or protein for amino acid sequences. So here I'm selecting nucleotide. Okay, and for example, we are typing here Pseudomonas aeruginosa. Pseudomonas 16 is RNA. And click on search bar. So here you will see multiple results. Click on the most relevant sequence based on your study. Make sure to check organism and sequence length. So suppose see here Pseudomonas aeruginosa PA96 genome, and it has 6444491 base pair, and it is circular DNA. Now look for the display setting or FASTA option. So you can see here FASTA option. Click FASTA and this will convert your sequence into FASTA format. Okay. Now here on right hand side you are going to see send to. Click on send to and select file and select FASTA sequence and click on create file. Your sequence now download to your computer. You can download multiple sequences at once. So here I'm going to show you the sequence downloaded. See here. So you can download multiple sequences by using the send to file option for search results. If you found this helpful, follow for more bioinformatics tutorials and AMR research tips. You can also ask which tutorial you want in comment box. Thank you.